Let me, before I show you the first slide, let me tell you the total story of heterothallism without showing any slide. और तब हम लोग slides के through जाएंगे, okay? So you be very attentive to what I am sharing with you. So hetero means different. Thallism is thallus related. So heterothallism here means different thallus related behavior. So thallism means thallus related behavior. We understand that? What is heterothallism? But what thallus related behavior we are talking about here? In this case, we are talking about sexual reproduction. Okay? So there are many fungi where sexual reproduction will be taking place only if two different type of thalli are present close to each other, and only between them sexual reproduction will be taking place. These two different types of thalli means what? They belong to the same species, but they are having slight genetic difference. Slight genetic difference with respect to their mating type. So if they will mate only with some other type, then this is one mating type, and the other one with which it is compatible will be called other mating type. Understood this thing? So some fungi display this behavior. This behavior is known as heterothallism. Point number 1. In our earlier discussion, when we were talking about asexual reproduction and sexual reproduction, we had spoken about that sexual reproduction is a slow process, is a complex process, produce the gamete, make the gametes fuse, make the nuclei fuse, carry out meiosis, carry out a spore production, so on. So it is slow, it is complex, plus it is wasteful. In the sense, what if after gamete formation, it does not get to fuse with the compatible gamete? Think of neurospora, which makes a spermatia. The spermatia will be produced in hundreds of numbers, and barely 10 of them will be able to fuse with the compatible hyphae. So what happens to rest 90? They just perish away. So that way it is wasteful also. Yet why is it so that all the advanced organisms display sexual reproduction? Because sexual reproduction gives you one advantage which asexual reproduction does not give you, and that is what? The possibility of genetic novelty. In sexual reproduction, when gametes unite, the gametes are most likely to be genetically different from each other. When they unite, the zygote has a mixed genetic profile, and then in meiosis, the mixed genetic profile will be distributed among the offsprings, and then offsprings will be differing slightly from the parents. That is why none of us is an exact replica of our parent. We have some features like our mom, some features like our dad, but do you not think that there are so many features which are entirely uniquely yours? Why? Genetic mixing. We are getting it? So that way is genetic mixing, which we call genetic recombination, is in a way advanced feature. And for that, slow, complex, even wasteful sexual process is all right. Organisms, as they accumulate genetic novelty, their adaptive features increase, and as the adaptive features increase over a long horizon of time, their chances of survival, even under changing environmental condition, is increasing, because new genetic features are giving you new abilities. These new abilities, you never know when they will help in your survival, right? So that way it increases your chances of survival. If the prime purpose of sexual reproduction is to bring about genetic mixing and genetic novelty, heterothallism is a superior type of sexual reproduction, because it insists that gametes must come from two different genetic types. You got my point? Because if the gametes are coming from the same genetic type, even their union is not going to give you that desired recombination for which you are carrying out sexual reproduction. But heterothallism insists that no, gametic fusion will happen only if the gametes are coming from two mating types. Mating types are different genetic types which are compatible to each other but incompatible to their own type. Means in Rhizopus, plus will not mate with plus, minus will not mate with minus, but plus and minus can mate with each other, hence mating type. So this is a superior type of sexual behavior. Agreed? Okay. This is a type of sexual behavior where even though on the same thallus the reproductive structures are forming on the same thallus, okay? So what is this? This is antheridium. Well, what is this? This is ascogonium. Even though they are growing on the same thallus, if between them mating is not taking place, this is what? This is true heterothallism. Okay, this is true heterothallism. And don't draw anything. बाद के स्लाइड्स में ये सारी चीजें ड्रॉ की हुई हैं, मैं शेयर करूंगा आपके साथ. ओके, बट जस्ट ट्राई टू अंडरस्टैंड द स्टोरी. दिस इज ट्रू हेटेरोथैलिज्म. देन देयर आर सम फंजाई. रिपीट कर दूं? बस एक मिनट रुको. मैं कह रहा हूं कि देखो ये एक फंगस है जिसमें एस्कोगोनियम और एंथीरिडियम एक ही थैलस में है, लेकिन इनके बीच अगर मीटिंग नहीं हो रही है तो दिस इज ट्रू हेटेरोथैलिज्म. कि देखो दोनों पास में है, देयर इज एवरी पॉसिबिलिटी कि इनके बीच मीटिंग हो जानी चाहिए, फिर भी नहीं हो रही. दिस इज ट्रू हेटेरोथैलिज्म. देयर आर सर्टेन फंजाई जिनमें एक थैलस में ये बन रहा है और दूसरे थैलस में एंथीरिडियम बन रही है. Right, two different thalli. Is it true heterothallism? Perhaps not. Why not? Because here you are sexually dimorphic. Are human beings heterothallic? No. Because human beings, or for that matter any mammal, is sexually dimorphic. Male is separate, female is separate. Heterothallism you will apply when there is no sexual dimorphism. Male is not separate, female is not separate. They both are present on the same fungal structure, yet they are not compatible. That is what? Heterothallism. I hope you understood this point very clearly. Okay. So, पुरानी किताबों में इसको भी लोग heterothallism कहते थे कि this is morphological heterothallism and this is physiological heterothallism. Today, for modern day mycologists like Alexopoulos, James Deacon and all that, this is the real heterothallism. They are present, they are close by, but they are not compatible. You got it? यहां तक समझ में आ गए? Okay. Heterothallism, yesterday as I was telling you, was discovered by A.F. Blackesley in Mucor. Yesterday we saw the life cycle of Rhizopus. So Rhizopus and Mucor life cycle is very similar, and in both of them we see heterothallism. But heterothallism is present in other fungal phyla and even in the phyla of fungi-like organisms. So practically in every phylum you will find a couple of members displaying heterothallism. That is about occurrence. So you will find it in Chytridiomycota. Zygomycota में तो डिस्कवरी ही हुई है, एस्कोमाइकोटा में तो पड़े हुए हैं, ठीक है. फिर बैसिडियोमाइकोटा में भी है. फंजाई-लाइक ऑर्गेनिज्म की अगर बात करो तो वेरी डेफिनेटली इन ऊमाइकोटा. All of these groups, they display heterothallism. Understood it? And the operation is the same, that on between two different thalli, sexual fusion will be taking place. Agreed? But now comes the question that how heterothallism is controlled. So heterothallism, like most of the features, are controlled by genes. In some of the organisms, heterothallism will be controlled by single gene. In some of them it will be controlled by two genes. If it is controlled by single gene, then there are two mating types. For example, in Rhizopus, plus minus. In Saccharomyces, A and alpha. In Neurospora, A and A, so on and so forth. So in this, when it is controlled by a single gene, then a single gene will have two alleles. Allele, you all understand? Allele is different form of the same gene. Okay, so one form is plus. If this gene is in the plus form, the fungus having this gene will be called plus, and if the gene is in the minus form, it will be called the minus one. Similarly, mat A, mat alpha, neurospora may be mat A and mat small a, so on and so forth. So if heterothallism is controlled by a single gene, there will be two mating types, and this is called bipolar heterothallism. Bipolar, why? Because there are two poles, plus pole, minus pole, A pole, alpha pole, capital A, small a. But then there are certain cases where you find two different genes together controlling heterothallism. In this case, imagine gene A and gene B control heterothallism. Gene A will have two forms, A and small a. Gene B has two forms, B and small b. Now, any one copy of gene A could be present in the same organism with any one copy of gene B. So, how many types of combination possible? Correct? How many mating types do you see? 4. What would you call it? Tetrapolar. As many mating types, that is the so-called polarity. So either you have bipolar or you have tetrapolar. Bipolar is going to be controlled by how many genes? Single gene, two alleles. Tetrapolar, two genes, and each one will have two respective alleles. Understood? Chalo. Now, beautiful slides. Right. मैं खुद ही beautiful कह रहा हूँ. अरे अब दिखेगा. Right. So sexual reproduction in fungi can be homothallic. In this case, what will happen? Parents are genetically identical. Can be heterothallic. Parents are genetically different. And that way, what? Offsprings display greater genetic variations. In that ways, it is advanced behavior. Do we not already know this? अभी तो पता चला था, right? So this is an advanced behavior. And why advanced behavior? Because it increases, मैं यहाँ add कर देता हूँ और मैं ये तो आप लोगों के साथ share करूँगा ही, because it increases genetic variations. And if it increases genetic variations, it will be causing evolutionary adaptations. Understood? So that is how it will be. ये देख लिया ना कि क्यों है ये advanced behavior. अभी there is no need to write. You have to make it, but when I share it with you. ठीक है ना? Any question here? No. हाँ बताओ. हाँ, sexual dimorphism include कर सकते हो, but you will have to mention that modern day mycologists, they consider only the other type, physiological type, as the true heterothallism. Understood? So if you are writing, what is it? Where sexual fusion occurs strictly between different mating types of thalli. Advanced sexual process, discovery by whom? A.F. Blakeslee. The year was 1904. Yesterday you asked, do I have to remember the discoverers? Only when it is landmark discovery, yes, not anything. And if you can remember the year, अभी difficult लग रहा है, लेकिन वो याद हो जाएगा. मुझे भी कम से कम 20 discovery year तो याद ही रहते हैं हमेशा. So like the discoveries which established DNA as the genetic material, then Calvin cycle discovery, Hill and Bendel discovery. So ये सब याद मुझे भी रहते हैं, आप लोगों को भी याद हो जाएंगे. So well, A.F. Blakeslee, and Blakeslee में C नहीं होगा. It is Blakeslee. कई लोग लिखते हैं black and then Esley. So no, that won't be right. So A.F. Blakeslee. Now you would want, does it matter? Yes, it does matter for the kind of people who get to become UPSC examiner. So 3 morals of the story. Now, do not underestimate yourself, do not overestimate your capacity to memorize, and do not underestimate the examiner. Okay, so this was A.F. Blakeslee's observations. Okay, pay attention to this. This has not been asked, but one day if the examiner throws a question at you, define heterothallism and explain its operation. यहाँ तक तो बहुत अच्छा है. Add a note on its discovery. So if you do not know this, you will be stuck. So you have to know this, and it is very simple. So this is A.F. Blakeslee's first observation. What Blakesley did, that he collected spores from a single sporangium in Mucor. Mucor is brother of Rhizopus. So from a single sporangium, he collected the spores, and then on a suitable medium, he grew these thalli. So these thalli grew. So he allowed it to grow, mature and all. So at the end, in zygomycota members, your expectation is that now I am going to see zygospore. Blakesley noted that no sexual reproduction occurred, no zygospore formation happened. 1904, very nascent days of science. Mendel's work was just discovered. No one knows about the genetic basis of things, right? You know now that Mendel's work, they remained anonymous to the world for close to 40 years. From mid 1860s until early 1900s, the world did not get to know much about Mendel's work. And then three different scientists, they co-discovered, rediscovered the Mendel's work. This will be a very fascinating thing to study in genetics, but later on. Okay, so कुछ भी नहीं पता, but just think, what a surprise it would be for Blakesley. What's wrong with these? Why aren't they forming spores? But this is how design of experiment thing plays out. Blakesley, अभी रुक जाओ, अभी बहुत जरूरी बात हो रही है. Blakesley thought, okay, I will take 2-3 different types of 2-3 different thalli. I will grow them together. So he grew different thalli. And mind it, आपके बोर्ड पे ये येलो और ये ब्लू दिख रहा है. In microscope, they all look the same. Yet Blakesley took a mixed population. He collected a spore and then he grew like this. And then successful sexual reproduction and zygospore formed. So what is it? Most of the people would have left here. Blakesley went here. And he discovered something. जो 118 years के बाद भी someone is taking his name. Boundaries. This is important piece of fact. Occurrence. So both modes common in all phyla of fungi and fungi-like organisms. Both modes means what? Homothallism and heterothallism. Both modes. Heterothallic proper fungi, Ellomyces and Blastocladiella in Chytridiomycota, Mucor and Rhizopus in Zygomycota, Neurospora, Saccharomyces pezziza, more also in Ascomycota, and then Basidiomycota में Puccinia agaricus. Agaricus is your field mushroom, edible one, right? Astilago. So they all are heterothallic. Plus in Umycota, fungi-like organism, Aclia, L छूट गया यहाँ पे, अभी लिखता हूँ इसको. Aclia ambisexualis, ठीक है ना? इसको रुको, ठीक से ही लिख देता हूँ, ऐसे तो कंफ्यूज हो जाएगा आपको. Aclia ambisexualis, तो वहां भी होगा. देन अगर आप पूछोगे कि इतना तो हो गया, तो कोई होमोथैलिक भी बचा है. देयर आर सम एग्जांपल्स फॉर यू जो होमोथैलिक है. एग्जाम में नहीं पूछा जाएगा, बट नॉनदलेस यू नीड टू नो कपल ऑफ देम. ओके, सो ये अकरेंस की बात हो गई. अच्छा, मोस्ट ऑफ द पीपल डू नॉट एडमिट, चूंकि ये बात पहली बार हो रही है, तो आई मस्ट टेल यू फ्रैंकली, In Asia, not only India, this is one of the most commonly misspelled word. Where is the misspelling? There are two sources of misspelling. बहुत लोग आर के बाद ए लगा देते हैं और बहुत लोग डबल आर नहीं लिखते. So the only correct spelling on the planet Earth, no British English, no American English, only correct English across the world is this only. So if it is happening to you, तो आज के बाद मत करना, ठीक है. O double C U double R E N C E, that is occurrence. Occurrence is a word that you will write at so many places in botany. This plant occurs there, this plant occurs there, so that is one. So ये हो गई बात, ठीक है. Types of heterothallism we have already discussed. अरे types of heterothallism we have already discussed. First we have got true heterothallism, where both sex organs develop on the same thallus but they are incompatible, right? That is true heterothallism. Any doubt around this anymore? नहीं ना? Okay. क्योंकि हमने शुरू में तो देख ही लिया है. And then second is what? Sexual dimorphism, diosy. That is two sex organs develop on different thalli. Do you want an example? Many species of Phytophthora, they show male separate, female separate. But where have we included Phytophthora? Homothallic box. So today's botanists do not consider this behavior as heterothallic. They simply say that this is sexual dimorphism, this is no true heterothallism. Agreed? Okay. Next one. Operation of heterothallism. Now this is for your examination. ये बहुत जरूरी है. ऑपरेशन ऑफ हेटेरोथैलिज्म जब बता रहे हो ना, तो देखो कि कितने मार्क्स के लिए पूछा जा रहा है. ठीक है, अगर 10 मार्क्स के लिए लिख रहे हो ना, जस्ट शो वन एग्जांपल, और वो एग्जांपल राइजोपस का होना चाहिए या म्यूकर का होना चाहिए. ओके, 10 मार्क्स में एक ही एग्जांपल दो, और वो एग्जांपल दो जिसके बेसिस पे डिस्कवरी हुई थी. राइजोपस की लाइफ साइकिल हमने करी हुई है, सो वहां से आप कर लोगे, राइट. अगर 15 मार्क्स के लिए लिख रहे हो ना, तो एक न्यूरोस्पोरा का एग्जांपल दो, अगर 15 के लिए लिख रहे हो. और अगर 20 के लिए लिख रहे हो, so then you have to give 3 examples. अगर 20 के लिए लिख रहे हो, so you have to give 3 examples. Do you know why you have to give 3 examples? And why these 3 examples only? Because we are taking one example from each phylum. आप पेजाइजा भी लिख सकते थे, आप सैक्रोमाइसिस भी लिख सकते थे, बट ये क्या है कि एस्कोमाइकोटा से लेके भर दिया. एक एगैरिकस दिखलाओ या पक्सीनिया दिखलाओ, एक एस्को का मेंबर दिखाओ, एक ये दिखाओ. और हमेशा याद रखना कि 10 मार्क्स में एक ही एग्जांपल, बिकॉज यू विल नीदर हैव टाइम नॉर यू विल हैव स्पेस. वन एग्जांपल इज गुड इनफ फॉर 10 मार्क्स. न्यूरोस्पोरा पे चले जाना. कल मैंने ड्रॉ किया है ना न्यूरोस्पोरा में हेटेरोथैलिज्म प्लस माइनस एंड ऑल दैट, बस उसको ड्रॉ कर देना. So here you have general scheme of heterothallism. You already know that there will be 2 types, mating type plus, mating type minus. Plus will be separately carrying out its asexual reproduction, and minus will be carrying out its own asexual reproduction. But when they are growing close together, mating type plus and mating type minus, then there will be plasmogamy. After that there will be dikaryotic. Depending on the fungus, dikaryotic phase can be long or short. Recall in Chytridiomycota and Zygomycota it's going to be short, and in filamentous escomycota इस तरह की बातें लिख दो, वो एग्जामिनर खुश हो जाएगा. filamentous escomycota लिखने का मतलब क्या है कि you are excluding saccharomyces. Remember, in Saccharomyces, the dikaryotic phase is short. So in filamentous ascomycota and all basidiomycota, the dikaryotic phase is long enough. Okay. So dikaryotic phase, then karyogamy happens, then meiosis happens. When meiosis forms spores, half of the spore will be plus type, half of the spore will be minus type. So plus type will again grow into plus thallus, minus will grow into minus thallus. They both will carry out their own asexual reproduction, but when their reproductive structures, sexual structures unite, because they are from opposite sides, they will be uniting correctly. Understood it nicely? Good. Rhizopus, we have already seen it, isn't it? Rhizopus life cycle we have already seen. So here notice that it is plus mating type, right? It is minus mating type. Towards each other they are making their gametangia. कल के diagrams आप याद करना. Then these gametangia are cut off. Eventually these gametangia they fuse together. ये pixelate हो रहा है, लेकिन अगर थोड़ा छोटा करूँ तो you can see it. Fuse together, karyogamy, diploid nucleus, then meiosis, and after meiosis spores will be released. Half of the spores will form what? Positive, half of the spore will form negative thallus. So अगर एक दे रहे हो तो राइजोपस का देना जो हमने कल ड्रॉ किया था. This is neurospora की life cycle. If you want to simplify it, then so in neurospora you can draw this figure. ये बना लो आप लोग. This will evaporate after the class is over. So so this is the structure in neurospora. So this whole structure is called archicarp. Okay. And then spermatia will be produced. Wait a moment. So and then so this is a spermatium. So spermatia will be produced by other mating type, hence in different color, right? And then it will be fusing together. So this is A type and this is small a type. Spermatia are the male gamete, and this is receptive hyper, that is the female structure. So between them there will be fusion. If you are running short of time, इतना भी बना दोगे, it will be good enough. ठीक है ना? Okay, चलो. So this is for neurospora. ये लिख लो that this is for neurospora. Okay, now तो अगर टाइम नहीं है तो डोंट मेक द एंटायर लाइफ साइकिल, बट उतना तो दिखाना ही पड़ेगा हमें. And this is for agaricus. So in agaricus what happens, that haploid mycelia, they go through somatogamy. Do we remember from yesterday what was somatogamy? Right? Simple ordinary mycelia, they fuse with each other. But again in agaricus, the fusion will be taking place between plus and minus only. So that is the heterothallism of agaricus. If I make another diagram, so in agaricus, this is plus type, and this is another type. All right, and then you can mark them as plus and minus. You can mark them as plus and minus, and then so यहाँ पे there will be an opportunity of somatogamy, and this is what? This is something we see in agaricus. Agaricus is what? A basidiomycetes edible fungus. Done? ये बना लिया आप लोगों ने? अरे some of the cells you can draw at home also. Some cells, some nuclei. So this picture is clear. Last part. Subtypes of true heterothallism based on what? Based on the number of genes which are responsible for mating type determination. So if there is one gene, okay, if there is one gene that is having two alleles, like in Saccharomyces, MAT A, so A and alpha, agreed? So only two mating types, Rhizopus plus and minus, Saccharomyces A and alpha, Agaricus plus and minus. So in such cases, what we will call it? Bipolar, something that we had studied earlier. And if there are, if there are two genes, four alleles, so A can have two, B can have two, as a result there will be four mating types, 1, 2, 3, 4, all the combinations. It will be known as what? Tetrapolar. Bipolar is much more common than tetrapolar. Bipolar is less common. So notice, so bipolar is simple, present in all Ascomycota, most common in other phyla also, ठीक है? Like Ascomycota में तो देख लो, Saccharomyces, Neurospora, Pezaiza, Rhizopus, Mucor, Puccinia, Agaricus. Puccinia and Agaricus are what? Basidio, right? Mucor is Rhizopus, and ये कहाँ के हैं? Zygo के हैं, right? And these are what? They are Asco. And then tetrapolar is complex and present in only some members of Basidiomycota. One example is good enough, Coprinus. That's all. And I'm going to share this handout with you. So well, that is about heterothallism. Advantages you already know, that it gives you greater genetic variation in subsequent generation, and if that is there, there will be greater range of adaptation, and if greater rate of adaptation is there, it is going to have a chance of better survival, right? So the more variety a species has got, the greater will be its chance of evolutionary perpetuation, that even under changing conditions, it will continue to grow. I hope you understood this. So that is all about heterothallism. Now you can do your homework, making your notes. I'll be uploading it straight away.